What if we could listen in on the secret conversations happening inside our own cells? What kind of stories do you think they'd tell? Well, today, we're going to talk about a technology that does pretty much exactly that. It's called RNA-seq, and it is absolutely revolutionizing medicine by letting us understand the code of life as it's happening.
So let's start with a really fundamental mystery in biology. Every single cell in your body, from a muscle cell to a skin cell, has the exact same DNA blueprint, the very same genetic code. So how is it possible that one cell becomes part of a beating heart while another becomes a neuron firing away in your brain? Well, the answer isn't in the blueprint itself. It's all about how each cell decides to read that blueprint.
And that one simple question leads us to a really powerful idea. See, the difference between a heart cell and a brain cell isn't the DNA they have, but which parts of that DNA are being used at any given time. It really all boils down to which genes are switched on or expressed and which ones are switched off. This whole process has a name. It's called gene expression. And honestly, thinking of it as a simple on-off switch doesn't really do it justice. A much better way to think about it is like a dimmer switch. In any cell, some genes are cranked way up, shining super brightly. Others are dialed way, way down, so they're barely even on. This precise tuning is what gives every cell its unique job, and it's what can make the difference between a healthy cell and a diseased one.
Okay, so the next logical question is, how on earth can scientists measure the setting of every single one of these thousands of dimmer switches all at the same time? Well, that's exactly where the technology of RNA-seq comes in. It lets us take a complete genetic snapshot of a cell at one specific moment in time.
So how's it work? Well, at a high level, the process is actually pretty elegant. Scientists will start with a biological sample, let's say a tiny piece of a tumor. They then use this incredibly powerful machine to read or sequence all the active gene messages inside those cells. These messages are made of a molecule called RNA. You can think of it as a working copy of any gene that's currently turned on. Then the machine translates all of those biological messages into a massive amount of raw digital data.
And when I say massive, I mean it. A single tiny little tissue sample can spit out over 50 million individual data points. This isn't just a stream of information, it's an ocean, a giant sea of numbers that by itself is completely meaningless.
And this is where the story takes a really fascinating turn. The challenge is no longer just biological, it's a data science problem. We've got all the information, but now we have to make sense of it. How do we find those crucial signals of disease that are hidden deep inside that ocean of statistical noise?
So the first hurdle we have to clear is something called normalization. Here's the deal. Sometimes the sequencing machine just happens to read one sample a lot more than another. Scientists call this greater sequencing depth. This can make it look like all the genes in that one sample are way more active when it's really just a technical quirk. Normalization is the statistical process that corrects for that. It basically levels the playing field so we can make a fair apples-to-apples comparison.
Now, this next challenge is really interesting. It's called the multiple testing problem. Think about it. If you test, say, 20,000 genes to see if they've changed, pure statistics says you're going to find hundreds that look significant just by random luck. It's kind of like flipping a coin 20,000 times. You're bound to get some weird streaks that don't mean anything. We need a way to tell the real biological changes apart from these statistical flukes.
The solution is this really clever statistical tool called the false discovery rate, or FDR for short. You can think of it as a super sophisticated filter. It's smart enough to know that when you run 20,000 tests, you're going to get some false alarms. The FDR adjusts our expectations, and it lets us confidently pull out the genes whose changes are legit from the ones that are just, well, statistical noise.
Okay, so we've tamed the data, right? We've leveled the playing field, we've filtered out the noise. Now, the data is finally ready to tell us its secrets. And the way scientists visualize all this information is both beautiful and incredibly powerful.
First, we need a way to measure how much each gene is actually changed. For that, we use a metric called log2 fold change. It sounds a little technical, but it's really just an intuitive way to measure how much a gene's dimmer switch has been turned up or down. A value of +1 means its activity has doubled. A value of -1 means its activity was cut in half. Simple as that. It just tells us the magnitude of the change.
So, we take that magnitude of change and we combine it with our statistical confidence, that FDR value we just calculated, and the result is this single powerful chart that shows us everything, the volcano plot. Every single dot on this plot represents one gene, and its position on the chart tells us its story.
And reading this plot is surprisingly easy. The higher up a gene, the more statistically significant its changes. The further to the right, the more it's been turned up. The further to the left, the more it's been turned down. So the genes we really care about, the key players in our story, are way up here in the top corners. All these genes in the middle, they're just background noise. In one glance, we've gone from 50 million data points to just a handful of truly important genes.
This is all really fascinating, but so what? What can we actually do with this information? Why does finding these genes matter so much? Well, this is the part where the story goes from code to cure.
The real-world impact is just staggering. By comparing a cancer cell to a healthy cell, we can pinpoint the exact genes that are driving the disease. This helps us find biomarkers to diagnose things earlier, it helps us understand how a tumor might evolve, and, this is the big one, it helps us discover brand new targets for developing drugs. This is the foundation of personalized medicine, treating a patient based on their unique genetic activity, not just their symptoms. So, from one basic question in biology, we've gone through high-tech sequencing, wrestled with massive data challenges, used some elegant statistical solutions, and we've arrived at life-changing medical insights. It really leaves you with a final kind of tantalizing thought. Now that we have this incredible power to read the dynamic language of our genes, what profound mysteries of life and disease are we going to solve next?